Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about in situ hybridization, which is part of the IBMS Specialist Portfolio in Cellular Pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss indicative repertoire, knowledge requirements, competence requirements, reflective statement, evidence of achievement, Q&A suggestions, supporting evidence ideas and some helpful tips that can make your portfolio really good. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into the subsection. The portfolio offers indicative repertoire for this section that includes HER2, HPV, EML4 ALK fusions, EBV, immunoglobulin mRNA. Knowledge requirement is what it is expected of you to know and understand to demonstrate your knowledge is at the specialist level. Think about what knowledge you had as a Band 5 BMS. How would that differ to the knowledge as a Band 6 BMS? That specialist level of knowledge would allow you to troubleshoot, problem solve, verify, validate, help others and maintain high-quality service. Let's have a look at the first knowledge requirement, which is for you to know the principles of in situ hybridization techniques, including the use of fluorescence and chromogenic methodologies. What is in situ hybridization? How is this technique performed? Why is this technique used in histology? What is the difference between fluorescence and chromogenic method? What are their advantages and disadvantages? Consider sample preparation, fixation, permeability and pretreatment, denaturation, hybridization, washing, detection and visualization, evaluation, advantages, disadvantages, and application of FISH versus SISH methods. The second knowledge requirement is for you to know the role of the clinical application of in situ hybridization in diagnostic pathology. How is in situ hybridization used in diagnostic pathology? What are the clinical applications of in situ hybridization? Consider role in oncology and infectious diseases, pathogen identification, gene expression, amplification, rearrangement, translocation, fusion, disease monitoring or guiding therapy. Also consider the limitations and challenges of in situ hybridization. Competence requirement is basically a combination of knowledge, skills, and experience one needs to perform a job to a satisfactory standard and level. Your chosen evidence should demonstrate these competence requirements. The following competence requirements need to be met for this section. Let's explore the first competence requirement, which is for you to prepare cell and formalin-fixed, paraffin wax-embedded FFPE samples for DNA and RNA analysis. How would you prepare cell and FFPE samples for DNA and RNA analysis? What steps would you take and why? What problems could you encounter and how would you deal with them? Consider deparaffinization, rehydration if needed, permeabilization, denaturation, hybridization, detection, visualization, SOPs, method used like FISH or CISH, your training and competence. The second competence requirement is for you to stain tissue sections using in situ hybridization. How would you stain tissue section using in situ hybridization? What steps would you take and why? What problems could you encounter and how would you deal with them? Consider tissue preparation prior staining, stains highlighted in indicative repertoire and stains used in your department, SOPs, methodology used, control material, your training and competence. The third competence requirement is for you to select control material, maintaining appropriate audit trails. How would you select control material for in situ hybridization? How would you maintain audit trail? Consider tissue type and availability, control tissue storage, record keeping of control material, selecting correct control material, verification and validation, traceability and audit trail. The fourth competence requirement is for you to use appropriate microscopy techniques to visualize stained material. What microscopy techniques can you use to visualize material stained with in situ hybridization? Consider light microscopy, immunofluorescence, filters used, magnification, Kohler's illumination, digital pathology and AI. The fifth competence requirement is for you to assess quality in prepared sections. How would you QC sections stained with in situ hybridization? How would you know the staining technique has worked or not? Consider specificity of staining, uniformity of staining, presence of background staining, contamination or deposits, presence of nonspecific staining, signal intensity, UK NICA's assessment criteria, control material used, documentation. The sixth competence requirement is for you to clearly distinguish between positive, negative, and equivocal results. What are positive, negative, and equivocal results for in situ hybridization? How would you distinguish between them? Consider SOPs, methodology used, your training and competence, controls used, ratio, copy number, signal, characteristics, criteria, interpretation or resolution. The seventh competence requirement is for you to resolve problems associated with the demonstration methods, including root cause analysis, as appropriate. What types of staining problems can you encounter during in situ hybridization? How would you resolve them? What actions would you take? How would you communicate these problems? Consider investigation, problem identification, corrective actions to fix the issue, preventive actions to minimize problems from reoccurring. Consider possible problems like poor signal intensity, poor specificity or sensitivity of staining, artifacts like uneven staining, reagent contamination, effects of fixation, processing and antigen retrieval on quality of ISH staining, nonspecific staining, background, absent signal, or possible cross-reactivity. The eighth competence requirement is for you to dispose of waste reagents and residual material safely. How do you dispose safely reagents and residual material? What disposal risks are associated with those reagents and samples? Consider licensed contractors used by your lab, waste segregation, safety data sheet of reagent often state disposal ways, risk assessments, local policies, and regulations that are followed, health and safety risks associated with reagents like flammability, corrosivity, environmental toxicity. Also consider risks associated with disposal or residual material like human tissue. Each section also contains reflective statement which is asking you, the candidate, to summarize your role in the laboratory in the context with each section. When you complete all the subsections within this section, you will be required to write a reflective statement. You also needs to write a personal reflection on your training and why you chose your evidence. To prepare for that, write a smaller reflective statement for each subsection. What is your role in context of this subsection within the laboratory? What does your evidence demonstrate? Also write a smaller justification as to why you chose your evidence for this subsection. Also write what was your training like when it comes to this subsection. Writing a smaller reflective statement at the end of each subsection will then make it much easier at the end of the section. Each subsection also has evidence of achievement. This is a part where your training officer signs you off as competent and fit to practice as a BMS at the specialist level. Training officer will confirm that you can work in accordance with local SOPs relevant to this section. They will also confirm that you have answered questions set by them to test knowledge and skills relevant to this section. And they will confirm that your chosen evidence demonstrates your competence relevant to this section. You will need to be signed off as competent by internal assessor for this section. This can be your training officer, senior BMS overseeing this section, or a training lead. Questions should be set by the training officer. They need to be based around the knowledge requirements, relevant for each subsection. They should allow the candidate to demonstrate their understanding in each subsection. The questions should be specific and relevant to the candidate's in-house practice. They should also allow the candidate to demonstrate their specialist knowledge. If you would like to see specific example questions, please have a look at the video for trainers. Link will be included in the video description. These are some ideas for supportive evidence you will have to submit to demonstrate your competence in this area. Some more ideas for supportive evidence to demonstrate your competence in this area. Some more ideas for supportive evidence to demonstrate your competence in this area. I would also like to share with you some helpful tips that can make your portfolio look really good. Try to present the information in various formats such as tables, pictures, photographs, diagrams, flowcharts, graphs, or even videos, if you can. This nicely breaks down a lot of text that is usually present in specialist portfolios. Try to make it a little bit more interesting to the reader, which will be your training officer and eventually the assessor. Try to put yourself in their shoes. How would you like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture is worth 1,000 words. I will make a video. I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also try to use various types of evidence, not just one type, like case studies for each subsection. You could use experiments, case studies, annotated results, annotated photographs of you performing tasks, evidence of troubleshooting, annotated EQA results, nonconformances, validation and verification as comparisons of different approaches and their pros and cons, control material production, certificates of course attendance with reflective statements, annotated images or charts, and so on. Try to bring in your personality. Think about the ways you like to learn best. If you like experimenting, perform experiments. Like troubleshooting, perform troubleshooting. Choose something that demonstrates your understanding, knowledge, skills, and how you apply this in your practice. And one more thing, keep the competence requirements in mind when you selecting your evidence. Good evidence will tick off several requirements. And this brings me to the end of today's video. Hopefully you now have a better understanding about the knowledge and competence requirement for this section. And I hope you also have a better idea what evidence you can choose to demonstrate your competence. And my final take-home message? Keep those requirements in mind when answering questions and selecting your evidence. Bye for now.